Se retransmite porque atienden estudiantes de otros niveles también.
Sí, bueno, gente en general, ¿no? Sí, presencial. Se emite para otra gente, ¿no? Estudiantes están todos. Hola, ¿qué tal? Okay, so I think we can start. So welcome everyone. So I would like to start introducing myself, especially not for you because you know me, but for the people who is potentially watching us through the channel, or they will see us in the channel in the future. So my name is Juanjo Nogueira. I'm the Spanish coordinator of the Master Studies on Theoretical and Computational Chemistry. And within the master, we have been organizing an activity for the last 2 months. The students have been organizing this activity because of the International Day of Women in Science. And in this activity, the students have chosen a prominent female scientist on the field of computational chemistry. And so they have been prepared a kind of talk highlighting her fantastic research. In a minute, they will introduce Petra Inhofe. So Petra was the selected scientist. So before the presentation of the students, I would like to say a few words. So first, Petra, thank you very much for accepting our nomination and invitation. It is great to have you here. So thank you for coming. And thank you very much also for the contribution you are doing to the field, especially in the simulation of biological, in the field of simulations of biological systems. Later, Christina and Sujel will talk properly about maybe a short overview about your research. And also, I would like to thank you, the students, because you have been, let's say, you have been doing a very good choice. I agree with the selection of the scientists, and thank you for the work. So very well done. And yeah, I don't want to steal more time, so maybe we can start. I would like to give the floor to Cristina, Ramos, and Sujel. So as I said, they will introduce properly Petra Inhofe. and then we will have the talk from Petra. So I think we can start. So Cristina, Sujel, the floor is yours. I will share the screen.
So, good morning, everyone. Welcome to all the people who is inside and also online. Juanjo, thank you for your kind introduction. We are really excited to represent all the students of the M1 Erasmus Mundus Theoretical Chemistry and Computational Modeling Master. Okay. We had the pleasure to be here today celebrating the Women's and Girls in Science Day. And we think that this is an important issue because, as you know, the gender gap in the scientific field is still really big, and we have to work on that. So today, we have Petra Inhofe here. And I had the pleasure to meet Petra in Congress last year in Benicàssim, and I found her job extremely interesting, and I think that it can contribute to our development as researchers, especially for all the women here, but also for all the men. And the thing is, why we choose Petra for celebrating this Women's and Science Day, Women's and Girls in Science Day. So, well, we first start traveling along his career. He started his studies, the bachelor and PhD in the Heinrich Heine University. Then she done a postdoctoral fellowship at the Max Planck Institute, and she started her career as a chemistry or in the chemistry field as an interim professor in the Heidelberg University. Then, as you can see, she has worked along Europe in several institutions and universities until she is did her application for as a professor at the Friedrich Alexander University, at the Department of Chemistry and Pharmacy, and also as a technical director of the Computer Chemistry Center.
So I thank you, Cristina, for introducing Petra Inhofe. So I want to talk about a little bit about her research and how important her research is, and a little bit about women in science. So she has extensive publication in prestigious journals. As you can see, these four are examples: Nature Communication, Chemical Physics, then the Journal of Physical Chemistry B, and so on. And her research span different kind of research areas from dynamics of DNA, classical molecular dynamics, and vibrational in phenol. And that showcase her expertise in the field and the impact she's having on the field. So next I want to talk about women in science. Like, why do we need women in science? That's the question when we talk about. So is it about like in the future we want to say that okay, our field has equal number of males and females researchers? I say no. It's not because of that. Because women brings a unique approach, insights, and problem-solving skills that we need to enrich our scientific field, and that will give us impactful theories in future. And I want all girls to look at Petra and see what a woman can achieve in our field, and I want you all to follow her footsteps, and in future, maybe you will become next Petra Imhoff. So thank you for all your kind attention.
And also thank you, Petra, for letting us immerse in such a scientific career. It has been a pleasure. And welcome here. And thank you for accepting our invitation.
So it's not Christina, it's Carmen, sorry.
Well, the presentation's being changed. Thank you very, very much for selecting me. When Franco contacted me, I think the first few things I said is, I don't know why you chose me. I still don't know. Not that you did a bad job, but I would have been able to name quite a few other female computational chemists, scientists instead of me. But yeah, I'm very honored and very grateful. I'm probably blushing now. Yeah, so thank you very, very much. I'm particularly honored. I will be presenting some of the work I've been doing throughout my career, but it's not the most recent. It's the stuff I like to talk about most. I presented some of this in Benny Kassem, so forgive me for showing that again. And people have seen me earlier because it's the favorite topics and the stuff I present most often when I give a taste of what we're doing. The title is very general: Understanding Recognition, Reactivity, and Regulation from Molecular Simulations. It's meant to show you, hopefully, what can be done with molecular simulation, and not so very much of the background or the theory behind. You're having lectures anyway on that. Still, well, I can't help it, I'm a teacher. I have two lectures, two slides showing you something. Computational chemistry can be understood as, this is what I present to colleagues who are not in the field, so you are computational chemists already, so you know this. Can be understood as you start from an experimentally available structures, especially in the field of biosimulations, or an AlphaFold structure nowadays, which already shows how old this slide is. From this experimental structure, you make a model, or it is usually also a model, which you have in the PDB. The model is atomic coordinates. And if you want to compute something, you need a potential for that. You need an energy function. It's either an empirical potential, it's a quantum chemical, quantum mechanical potential, or a mixture of both for pure memory. Examples are here. So this would be the classical potential with all these bonded terms and so forth. Or you solve Schrödinger's equation for maybe only the electrons and have the nuclei still classically, or you even do quantum dynamics, which you learned today. I just figured out. And with this recipe, you then run optimizations to get best structures, best pathways, optimal structures, or you do molecular dynamics simulations, classical trajectories. These are the two here, which give you a time series of positions and momenta, and also gives you statistics, which you should also have covered. And from this statistical analysis, one can, and the analysis of the trajectories, you can learn many more things. You can learn what is the most probable conformation, what is the most probable binding site. You can also get time correlation functions, so what is the lifetime of a certain thing. And then looking a bit differently on things by applying graph theory, you can also use get this data and convert it into a kinetic model, kinetic theory. You can also get communication and signaling pathways. And all this is what we're doing and a bit more, and apply to questions like how is a DNA repaired, or how is a damage recognized and repaired? How do fluorescent proteins work? It's not very much to talk about today. How does an enzyme function? How does a signal is transduced from ligand binding to receptor through the membrane to the G protein coupling or something? I already talked about that. So this is the one slide on molecular dynamics simulation, which I can happily skip over because it had an entire lecture on this. You solve Newton's equations of motion and some potential. This is the famous equation here. You get a time series of positions and momenta. And if you use a thermostat, so if you have temperature in there, you get the famous Boltzmann equation. So you get probabilities, or you get free energies if you convert that. The example here is on a bead and spring model just to simplify things and for teaching purposes, because that can be solved analytically. In the computer, of course, you do this numerically. And for proteins, for example, it's not only one bond, but so many that you have to do it numerically. So that's the recipe there. And this definition of a bond as a spring doesn't work anymore if you want to do chemistry, if you want to break a bond and form a new one in your simulation, or if you have a huge change in electron density, then you need to resort to things like quantum mechanics, because the old spring is not equal to the new spring. The old bond is not equal to the new bond. Well, that's shown here. So we do quantum mechanics, but you can't afford to solve a Schrödinger's equation for entire protein. So the trick there is combine the two worlds. Quantum chemistry for the region of interest, where the chemistry happens, where the electrons shift their positions most heavily, and do molecular mechanics, imperial functions outside, combined to QMMM, where the slash is causing most of the problems, I tend to say. And using these, basically, these approaches, we generate the data, which, after careful analysis, hopefully gives us some insight into, what did I say, reactivity, something, and regulation. Okay, I have 3 stories for you. The first one is very, very short, simply because I promised something with photoactivation. What I show you here is the fluorescent protein EOSFP. It's a relative of the even more famous green fluorescent protein, which some of you may have heard about. So green fluorescent protein has its name because it has this shiny color. So does EOSFP. They are very similar, and the chromophore, so the thing which makes the whole thing shine green inside this green area, is the same as in the green fluorescent protein. However, the environment is a little bit different, and EOSFP can be converted into a red fluorescent protein. If you shine blue light on it, it becomes red fluorescent. That's not convertible though, because something is damaged in this protein. So from a intact green protein, it's now broken red protein. What's happening, this has been solved by crystal structures, not by us, is there's a break of one bond here, so one residue is chopped off, which allows the pi system to become elongated. So instead of this single bonds here, you have a double bond, and then this is a larger chromophore, making it a red fluorescent protein. The whole thing works, as I said, with blue light, 400 nanometers roughly, and at low pH. And the question we asked back then, as I told you, it's also a bit of a historical talk, is, well, what's actually the protonation state of the chromophore? Because you can't see those in the crystal structures. And there were 3 variants being discussed. Actually, there can be 4, but the 4th is so irrelevant that we discarded that. One of them is a neutral chromophore, shown with N here, where you have a proton sitting at this phenyl ring, or this former tyrosine, the anionic form of which is without this proton here, so O- over there. And then there's a zwitterionic form where you do have a proton, but not sitting at the oxygen atom, but sitting at this nitrogen atom of the imidazole ring. They absorb at different wavelengths, or at least for the neutral and the anionic chromophore that has been measured experimentally. For the zwitterionic form, there was no information available. And we started off by doing pure quantum chemistry, so having only this chromophore, optimizing that on MP2 level, it's not shown here, and then testing various methods to calculate excited state energies of this optimized structure and compare it with the experiment. So one is time-dependent DFT, then there's CAS MP2, so high level, and then there are 2 intermediate choices, which is a semi-empirics, but with a multi-reference character to it. I'm not going into too many details. The bottom line here is the semi-empirics with multi-reference does a fair enough job. So, lucky us, we could use the cheapest method, being the most accurate, to also apply the whole thing in the protein. Remember, QMMM is, well, a little bit chromophore only, or a little bit more quantum mechanics, and the rest is empirical energy function. So what we did then is we calculated the chromophore in these 3 versions in the protein, and we ran classical MD simulations with the empirical potential first to get sampling, so, or the wibbling of the protein around the chromophore, so to have a bit of a statistics of the environment, and then took snapshots of those and calculated excitation energies. That gave us these calculated spectra here. So these bandwidth are with a bit of broadening of the lines from the sampling of the many different conformations in the protein and calculating the excitation energy. So the blue stuff here is the neutral, the green is the anionic, and the black is the zwitterionic version of the chromophore inside the protein. The dashed and dotted lines are small changes in the setup of the protein around, having a proton on another residue nearby or not. And the shades are the experimentally measured absorption spectra at different, well, at neutral pH actually. So this is clearly the anionic form, but experimentalists knew that. And this is most likely the neutral form, or very likely the neutral form, which agrees to what the experiment told us with the pH dependence. So we can clearly say that at 400 nanometers, it's the neutral species which absorbs. So the photoconversion is initiated by the neutral species, which makes sense because it works only at low pH. At higher pH, pH 7 or so, the anionic form dominates, and you won't absorb anymore at 400 nanometers. And why the whole conversion doesn't work with an anionic form would be yet another talk. Well, another group has solved some of this. We're still working on that, I must say, because right now the answer is it actually does work with the anion, but it has a completely different mechanism. So spoiler, maybe in 2-3 years' time I can tell you about that. Different story. So the photoactivation partly now. I have to talk about DNA repair. I can't help it. This is one of the major things we're doing. This is a busy slide already. The DNA repair I'm talking about today is done by an enzyme called thymine DNA glycosylase, TDG. What does this enzyme do? Or what has to be repaired? So in an intact DNA, you find these 4 nucleobases, G, C, A, and T. And in a Watson-Crick pair, so you have a C cytosine paired to a guanine. C, G. You would have a thymine paired with the adenine. That's the way the DNA encodes the genetic information of the DNA, and the second strand, the replication, is then based on this matching, this Watson-Crick matching. Imagine now you have a wrong nucleotide in there. So the C is not a C, but one of the other 3. You will have a wrong nucleotide in the replicated DNA that causes a mutation in the proteins being produced that may cause a disease. So DNA has to be repaired. Wrong nucleotide can happen from when the DNA is synthesized, but it can also happen spontaneously. If a cytosine is damaged by a spontaneous deamination, it becomes a uracil. That's a building block of RNA, so maybe not so difficult or not so difficult to recognize, it still is. If the cytosine is a methyl cytosine, so one thing with a methyl group over here, and these things occur, this is an epigenetic marker, so some cytosines are marked being something special, methyl groups. This is deaminated, it becomes a thymine. And this happens while I'm speaking here to your DNA. Don't worry, TDG is also working while I'm speaking here. Every now and then, methyl cytosine becomes a thymine. So this thymine is a building block of the DNA, but it's then at the wrong position because it's now opposite guanine. So this X here, for example, being a T opposite the guanine, that's wrong. Because if the DNA with a T in is read out, the next DNA would have an adenine there. Hence, you have a mutation. TDG, thymine, DNA glycosylases, recognizes the thymine is wrong, flips it out of the helix, that's encoded here, so, and chops it off. So it hydrolyzes the bond to the base, and then another repair enzyme puts in the correct base again. This is a crystal structure of the thymine base in the active site of the protein. This is a little bit of the DNA. Here's a snippet of the protein coming instead. And the job of TDG in recognition is to discriminate the thymine, the wrong thymine, so the TG mispair, from a correct thymine, TA, or from a CG pair. And all these other colors I talk about later, so you won't. So how does thymine DNA glycosylation to do this job. Well, we set about and ran molecular dynamics simulations, classical ones, from only DNA first, thinking of maybe there is already enough difference between TG-mis pairs to TA pairs or CG pairs. And indeed, yes, there is. So I spoil that. What we did is, because the base flip is so important to get the thymine out of the DNA helix into the active site, maybe it's more difficult for a proper paired T than for the mispaired T. And that is indeed the case. What I show here, this is the free energy profile of the so-called flip angle, so of the base being flipped out, forced in the simulation. That's a biased simulation, adaptive biasing. Well, I don't know if you know this enhanced sampling technique, adaptive biasing force. So pretending the protein is there by just applying the force and pulling the base out, and then reweighting this to get the actual free energies. And the higher the free energy, the more difficult it is, or the less probable. And you can see here that the red curve, so the one for the mispaired T, is much, much more shallow than for the other two pairs. So it's much easier for this mispaired thymine to be flipped out. Aha, okay, riddle solved. Yeah, partially. So what would that mean? TDG could run along the DNA and see whether there's a flipped out thymine signaling, hey, chop me off. Thinkable. However, the probability for this thymine to flip out spontaneously is still too low to rely on that. Recall our genes are at stake, yeah? So you want it repaired as good as, recognized as good as possible. Maybe too long to wait for it. The alternative, or the extreme opposite, would be an active mechanism. So again, protein running along the, yeah, this being the DNA, this protein running along the DNA, and testing every base, or at least every thymine, whether to flip this out. Also something which is maybe not too likely to happen, because that will also take a while. Well, as very often the truth is in between, or we think the truth is in between. So when you look at the DNA alone, again, without the protein, and compare the three situations, mispaired thymine, TG, red, paired thymine or paired CG, there's a second state in the mispaired case, shown here. This is a sort of properly paired TG, so as good as it can bind to the G. But there's another state where it has only one hydrogen bond. When you project it onto the opening angle or the flip angle, you see the second state. So there's a second free energy minimum, which doesn't exist in the other cases. So the green and the black control don't have that second state. So it is open, or a bit more open by itself, even without forcing it. It's less probable than the closed state. If we run the same exercise now with the protein bound, things still flipped in, now the second state becomes the more probable one. The free energy minimum being lower in the open than in the closed state, much more in the flipped out. And nothing happens, or not very much happens, for the proper pairs. So the protein, the mispaired T has a tendency to open already, and the binding of the protein stabilizes this state, so makes it much more probable. Recognizing there is something wrong with a small displaced thymine and stabilizing this state. So helping it more halfway out, and then the actual flip is much easier. Our take on the mechanism here. And when you then look at the interactions in the complex of the flipped out thymine, there are many, again from non-biased simulation, there are many indicators that the thymine is much better accommodated in the active site than the cytosine is, okay. But also the thymine with the G on the opposite strand is much more favorable than the TA state. I don't want to read all the details here. So there's larger occupancies for hydrogen bonds, that's better accommodation. The active site is much more wobbly with a methyl cytosine around. And you can also quantify this we did by free energy perturbation calculations to calculate how much more it's probable, or what's the higher binding affinity for cytosine versus thymine. I must admit that the error bars are rather huge, but it still shows points into the right direction. Okay, we know that TDG can apparently discriminate the mispair T, but how does it do its chemistry? To look at this, we ran QMMM calculations. So again, combining quantum chemistry with molecular mechanics, so with empirical potential. So small core is few atoms are treated quantum chemically to have electrons very flexible. We can break and make bonds. The rest is potential and an empirical energy function. And what we ran there is scans of several possible ways to how the mechanism could occur. And on top of that, we ran umbrella sampling to get free energy profiles. I think you've heard about this method. So what's the result? There's the best mechanism, or one of the best mechanisms, is a so-called direct, or we call direct mechanism, which works as follows. It's a dissociation step first, so the base leaves. So you have this CN bond broken. The base is chopped off from the sugar already, so everything done. However, you need to heal it a bit. It's a substitution reaction for those who remember a little bit of chemistry. You need a water attacking as a nucleophile to replace the bond with the base. So CN bond dissociation first, highest barrier here, then a little bit of reorientation, a water attack, the base rotates, and finally a proton transfer from the water molecule to the base. So you have an OH at the sugar, and you have one hydrogen at the base in the end. I said we have more than one mechanism. This is the second mechanism. Essentially the same. You have a high barrier for CN bond dissociation. So the base leaves the sugar. Cut. We have a water coming and attacking, so coming close to the sugar. The base reorients, so something like that, such that the proton transfer from the water can take place and the water can finally bind to the sugar. And we have now this proton here and the OH at the sugar. Why is this a second mechanism? Well, first of all, the barrier is lower. Second, it's called histidine mechanism for a reason. The whole thing is assisted by a preliminary, by a proton transfer from a histidine residue nearby to the base before the actual dissociation. If you pay close attention, we have a proton sitting here, which wasn't there in the other mechanism. And this proton comes from a protonated histidine. So in a two-step hop-hop mechanism, the base is protonated, making it a better leaving group because now it's neutral. CN bond dissociation and all the stuff before. And then, of course, in the end, you need a reprotonation of the histidine to complete the mechanism. So it looks a bit more complicated. The essential steps are the same, but they are easier. Easier in terms of barrier being lower. This is a schematic summary of what I just showed you of the two mechanisms. So the upper part is, these are the intermediate steps. The numbers here are the energies. The red numbers are the barriers, the activation energies. So we have 18 kilocalories per mole for the first step for the dissociation. And in the histidine mechanism, it costs us 12 kilocalories per mole. So that's more probable because it has lower energy. It has more steps, but fine. Why does it have a network of reaction pathways? Well, I have a proton transfer before the actual dissociation in this lower histidine-assisted mechanism, and then a proton transfer at the end. I can imagine that this proton transfer also happens at every other stage. So here, here, here. And in fact, yes, we calculated all those variants. So proton transfer before the dissociation, proton transfer after the dissociation, proton transfer after water attack, proton transfer, you get it. Hence connecting the two pathways and the calculated barriers with those. And figuring out all the energetics with which they are marked here, well, in numbers and in the thickness of the arrows. And along the thick arrows, this is the probable pathway, the reaction can now occur this way via DE, proton transfer back, this way or the other. So everything's rather similar or not so much important once the CN bond has been cleaved. And this has to happen, or is more probable with the proton being transferred first. So hence we have a network there. The histidine is important to facilitate the CN bond dissociation because it donates the proton. It's basically the catalyst activating everything. Now has the lower barrier. However, well, the experimentally measured rate translates with transition state theory to something of roughly 20 kilocalories per mole. And we say our best pathway, the most probable pathway, has 12. And the other one is 18. So maybe it's the other one. Okay. We went back thinking, what's the role of this histidine? Can this be true? Well, calculating things with a not protonated histidine and getting barriers for only the CN bond cleavage step, shown in the middle here. If you do not protonate the histidine and there is no proton helping the dissociation, you have a very high barrier. That's so high it will never happen, or almost never. If the histidine is protonated but is not involved direct mechanism, we have this almost 20 kilocalorie. And with the histidine being protonated and donating the proton back, this is the most favorable mechanism. We can't help it. But we have an explanation for that. The histidine is not necessarily protonated throughout. So we ran calculations of the probability for the histidine being protonated, the pKa value, from 3 different sets of simulations. So the histidine being in a protonated form, sampling the conformations, computing pKa, and in the neutral form. And the pKa is always 7 plus something. Normally, histidine has 6 point, I don't know what. So histidine is very likely to be protonated, but not completely sure. And if you do the math, or roughly, you get out that this conditional probability of the histidine being protonated, and only then it runs via the probable mechanism, fits nicely to the numbers we get out. So the fast reaction occurs only when the histidine is protonated. And then still, of course, the enzyme has both choices, going the direct or the indirect way. However, the histidine-assisted way is the one which explains a lot why thymine is being cleaved and not the cytosine, because the proton transfer to thymine is possible. The cytosine has an NH2 group, which won't accept the proton. So TDG has 3 recognized thymine. First of all, it's more wobbly, it's already a little bit out. It's much better accommodated in the active site. And once it's there, it's also the one which can accept the proton, and hence directions much more easily or at all possible as opposed to cytosine. I don't know how I'm faring with time.
Sorry.
I do have time. Then I do show you the second story of TDG. And then there's a third story of something completely different. I told you there are more colors here. So thymine DNA glycosylase not only cleaves thymine, despite the name. There are other damages. Once you have a methyl cytosine, this can be oxidized. There's another enzyme doing this actually, and the idea of this is getting rid of the methyl group, or people think it's getting rid of the methyl group, so of this epigenetic marker, and introducing further modifications of this. The first step of oxidation is converting the methyl group to a hydroxymethyl group. First year chemistry, methane becomes methanol. Second step is converting this alcohol to an aldehyde. So the methanol becomes formamide, or hydroxymethyl becomes a formyl group here. And yet more oxidation means we have a carboxyl group. HMC, FCCAC, these 3 colors. Interestingly, hydroxymethyl cytosine is not recognized by TDG either. Only these higher oxidized forms, formyl and carboxyl cytosine, are recognized by TDG to be damaged and chopped off. Okay, now everything I told you about how thymine is recognized can't be applied to a cytosine anymore, because this modification is on the other side. It doesn't affect anything on the interaction with the guanine. Still, TDG is capable of discriminating the two. And it still does base flip and so forth. Just to show you, we ran the simulations of the free DNA and of the DNA in complex. We calculated flip angles for all the 4 different forms. We ran adaptive biasing force to flip out the base of the DNA. It was such a nice story before, and now with these oxidized forms, maybe they are all pretty much alike. And the truth is in the chemistry, I must admit it is, but I don't show you today. But there's a bit of discrimination before that. So is there maybe recognition in the extra helical complex? I told you that methyl cytosine is not so well accommodated compared to thymine. Now let's compare methyl cytosine with the oxidized forms. So on the right hand, we have methyl cytosine as before, and then we have the higher oxidized forms over here. This is a snapshot from the MD simulation overlay to the crystal structures for checking, okay, are we somehow completely wrong or not? They all look rather nice. Some nicer than others, but okay enough to say, come on, there's not, thanks to one of those oxidized forms, this is completely different from the crystal structure, so completely wrong. Okay, if we then do quantitative measures, or more quantitative, like seeing how often we have interactions or which interactions and what are the numbers to that, thankfully we see a difference. One is very easy to understand. There is an important hydrogen bond between the tyrosine residue and the oxygen atom of the oxidized form. So this tyrosine residue has a backbone NH, which makes a hydrogen bond to an oxygen of the bases in the methyl group. The methylated form doesn't have this oxygen, so it can't have the hydrogen bond at all. And for the other three, you have different probabilities, 0.9 something for the two forms which are processed, and much lower for the other one. Stronger binding here. We can also quantify this by interaction energies of the base with the entire active site. Much stronger for the carboxyl, this is because it's charged. Very strong for the formyl still, not so bad for hydroxyl, and much lower for the methyl. If we look at distances between the base and our famous histidine, we also see that the distances are shorter in the two forms, yellow and aubergine, eggplant-like here, meaning there's a stronger interaction there, maybe not so important because of the proton transfer being different. They're closer. The actual quantitative measure comes from then doing free energy perturbation calculations, so calculating quantitatively the free energy difference between a methyl cytosine being bound or one of the oxidized forms. And we did this in two variants of the histidine, so being a protonated and not a protonated one. We already know the protonated form is the one which is not active. And the ranking is very clear. So there's much higher preference, -6 compared to something for the oxidized form. So the binding affinity of the formyl and the carboxyl cytosine is so much higher, or well, not so much, it's much higher than for the unoxidized form. So it's simply bound so much better that the others don't stand a chance of being free. Sometimes it can be as simple or seemingly non-simple as this. So to summarize the DNA repair part, I showed you that TG mispair is sort of easy to recognize because it has several stages when it can be recognized: before binding, at the flip out, after binding. But also the oxidized forms can be recognized. They only in the later stages, once the base is in the active site and during the chemistry. And by this combination of molecular dynamics simulation and QMMM and quite some thinking, we could crack the mystery of TDG and how it repairs DNA. I hope you still have some energy. That's when, yeah, you'll never invite me for such a long talk. That's the other thing I love to talk about. There's so many more. Don't worry, that's the last example I have. Cytochrome C oxidase. Completely different story. Nothing with DNA. It's a molecular fuel cell. It's the membrane protein, part of the respiratory chain, also active while we breathe, while we speak. What does this enzyme do? It does chemistry. It makes water, like a fuel cell. It takes hydrogen and oxygen, burns it, and makes water. It's not directly hydrogen here, it's protons and electrons. They are converted into water, and the energy which is produced by this reaction is used to pump protons through the membrane. That's depicted here. So from the inner of the cell to the outer part against the proton gradient. So there are already too many protons outside, so it takes energy to pump more protons up. And this gradient, or these too many protons outside, are then later used by ATPase to produce ATP, to store the energy of your food actually into ATP for later use. Not something you can use in your cars, but your cells are doing that. The mechanism of cytochrome C oxidase doing this job, or this dual job of producing water so as to pump protons, is rather complicated and depicted in this cycle here. It goes via several redox steps, and each proton uptake and each redox step is associated with the proton uptake. Some of the protons are used for the water production, some of the protons are pumped. And what's known further about cytochrome C oxidase is it has, at least in this species, 2 channels for the uptake of the proton. They are named D and K channel, named after important residues in aspartate and lysine. This is how the protons are, through which the protons are transported to the reaction center. And one channel is only active in one half, the K channel is only active in the second half, the D channel is active for the chemistry. So for this whole thing to work, is the protons need to know where they are. The protein needs to know where the protons are, so to speak. Protons should be allowed to travel through one channel only when they're required to do so, for the chemistry or not. Otherwise, you have one step or one part of these steps before the other step is actually taking place, blocking the cycle. No, things are not regulated. So here we have the regulation part. So we first asked, how are these protons going through the 2 channels? Well, let's start with the D channel. What does this proton transfer look like? It's a Grottis mechanism. This is, I think, known to you. There's this relay mechanism. You have a proton going from one water molecule, let's say, to another, and then not this very same proton is continuing, but another one is going from here to the next, like when you run a relay, you give the proton to something. That is not a simple reaction coordinate. Overall, yes, you transport one charge from here to there, but it's not like, okay, you bias one distance. Maybe, and you don't know who are the players or the runners here, as opposed to a sports event. And you have to sample many possibilities. And our trick here is divide and conquer, or discrete path sampling, or transition networks. The idea is depicted here. You have a starting point, reactant state, proton being just outside. You have an endpoint, product state, proton being where the water is being produced. And you can imagine there are many, many intermediate states, just schematically shown here. And you can go either this way or that way, network of reactions. The underlying picture, this hilly landscape, is the energy landscape. People who are into hiking get the idea of, okay, you will not necessarily cross the highest mountain unless you're into mountaineering, then people love it doing exactly this, but you would rather go something more shallow. How do we do that? So you first choose the degrees of freedom you want to select. What's important? Of course, the proton positions are important. Maybe some water positions, side chains, which can rotate and do everything. And these are, in a clever manner, I don't go into too many details, sampled so as to generate several possibilities. So to combine all this. We have the 2 end states, proton sitting here, proton sitting there. They are known, we have the optimized structures. We do not know the underlying energy landscape, but we do know how to calculate it. That's meant by the recipe is known. Then we sample these degrees of freedom, shown here only in 2D because I can't represent any more, like one distance or another, proton positions, generate in a grid, for example, many, many possibilities. Then, out of those points, they are optimized such that only good structures are retained. So if we generated something where 2 side chains bump into each other, that is dismissed right away because that doesn't make sense at all. It has high energies. We only want low energies. So few of those intermediate states are kept. These become the nodes of a network. Then we see which of those are neighbors. In this 2D example, it's easy because it's just by Euclidean distance. In higher dimensions, it takes a little bit other measures also. And then we connect those nodes of the network, which are neighbors, and say, okay, they have an edge in between. And for those edges, we calculate the actual transition. So between two minima, which have been identified to be close to each other, we calculate the transition between these two nodes. Transition state search, as simple as it is. So we compute the barriers, but only of selected transitions, not each and every, and not the very big from here to there, but only these from one village to the next, if you will. This gives us, mathematically speaking, a graph or a network, the transition network. The nodes of the network are these minima structures. The edges are the connections. The edge weight, the cost for the edge, is the barrier in between. And on such a graph, then with a simple Dijkstra algorithm, that's a very, very well-known graph theoretical algorithm, we can find the best way for going from here to there. So this being our network, now schematically, it's not the one, with the calculated numbers, the best pathway to go from here to there is this purple one. This is the one where the maximal barrier is the minimal, the lowest one among all maximal barriers. So basically, if you compare all the mountains you have to climb over and all the different routes to go from here to there, you would probably, you will not cross the Mount Everest if you can avoid that. This also gives you a little bit more than the best pathway. It gives also the second, third, and so forth best pathway. And it is a simplified, discrete energy landscape representation. And I love to compare this to a public transportation map. This is Berlin. Well, it's not Berlin. It's the public transportation map of Berlin. So a simplified representation of Berlin. And if you want to go from Westkreuz to Ostkreuz, so these two stations, then the fastest way normally is this connection here. Then people who have been in Berlin know there's very often constructions or something going on, so you probably can't go all the way through. Say one node is blocked, you have to take the underground or something else to use another path, like here, second best path. And if the underground is also not working, actually today they're on strike, you would take a bus, but you can't do today either. But yeah, anyway, you have another detour, third best path and so forth. Yeah, so far for the analogies. The same can happen, of course, in your enzyme. Okay, this is the most probable one, but it takes this or that. So we get all these for free. So summarizing for the CCO, the best pathway, on the left-hand side, you see the network in the schematic picture. The proton has to go from down there to up to the reaction center. All these circles are possible minima in between. These numbers here are a code for what actually has changed. Don't worry about the actual code. And the red numbers are the energies. This is the very simplified chemical version of the best pathway. The proton has to go from this aspartate of the D channel to the glutamate up in the D channel. And it works by an opening of these two asparagines, so an asparagine gate. It's called opening, such that water can go through in between. Once it does, then there's a hydrogen bond chain formation. That's what HBC stands for, which allows the proton transfer along the Gratis mechanism. Hop, 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 hop. And then finally it goes to the product. And you can also see we have a second, third, and so forth best pathway. They're not too different. They all need this asparagine gate to open. So we found a pathway which also agrees with what other people have studied before. Regulation, I told something. Okay, communication, protonation dynamics. So how is this regulated? Okay, asparagine gate is important. Keep that in mind. What we did to understand better the regulation is we then ran molecular dynamics simulations with empirical potential of the cytochrome C oxidase system in different protonation states. So we had the proton here, there, somewhere else, simultaneous, parallel, not the proton transfer as such, but if the proton is here and can't go anywhere because of the bond potential, how does it look? Or how do things change? And the most important bit for the D channel is shown here. So one is a situation where the proton is still in the lower part, that's on the right-hand side, still has to travel through. And on the left-hand side, the proton is already where it should arrive. This is a snapshot, and you see already when the proton is down here and still has to go through, maybe you see this asparagine gate is open, so these two asparagenes are far from each other, water is through. You do have a very low probability for hydrogen bonds between the two asparagenes, so that's also a measure for that, as opposed to the situation when the proton has arrived already at the top of the channel, where the probability for the hydrogen bonds between these asparagenes is very high, corresponding to a situation where this gate is closed and no water can pass through. And no water can pass through can be quantified again. So this is the number of water molecules, or maybe these plots are projections of the number of water molecules. So like these rain clouds, you see. Now there's fewer waters here than over there. And the water molecules are necessary to pass the proton. So the D channel of CCO itself makes sure that once the proton has arrived in the upper part of the channel, everything is closed and no proton can come through again. Clever, isn't it? No second proton coming and disturbing the cascade of all the other things in the cycle. So how does it do that? Or how comes that once the proton is up there, things are closed down there? Well, hydrogen bonds are the key here. Okay, this looks more complicated than it actually is. Again, I show you an open and a closed situation. Sorry, I swapped the sides. So the closed side is on the right side and the open is on the left. More water molecules than here, perhaps. If you look at the hydrogen bonds and their directions, so hydrogen bonds have a donor and an acceptor, then this asparagine is with its donor, the NH2 group, pointing up, because up there is the glutamate being an acceptor, charge residue. So all positive charges align more or less up there. And the probability for finding a path, so a connection along hydrogen bonds from the down to the up of the channel, in this situation where the proton is not yet at the top, is very high. It doesn't exist in the situation, or almost doesn't exist in the situation, where the proton is already there. No connections fully through. However, in this scenario, there is a connection from the glutamate up there down to the asparagine of the gate. And this perspective of going down is actually what you need to see in these arrows here. So the glutamate has a proton now, is the hydrogen bond donor, which points down, 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 to the asparagine, which now has its acceptor part up, the oxygen. And the donor part is now capable of making the hydrogen bond to the other asparagine, closing the gate. So structurally, it's all there. If you do this exercise, not for only 2 states, but for all the 16 possibilities we tried, and compare the graphs, again, networks of hydrogen bonds, they're direct included with each other, and calculate the similarity, submeasures, you see the following, depicted here in this matrix. This is so the 16 variants. 1 means the proton has already arrived. It shouldn't be going through. And 0 means it should still go through. Red means it's very similar. Blue means it's very unsimilar. So whenever you compare a 1 with a 0, you have a blue or very blue or dark blue, completely different, so not similar situation. And if there's a 1 with 1, so glutamate already there, they are highly similar. So the hydrogen bond structure is dependent on the protonation state of the glutamate, basically only on that. That's the major decisive factor of in which directions the proton are looking, so to speak. So the D channel of CCO regulates itself by making sure that the hydrogen bonds are in the direction the proton has to travel, or traveling down, it cannot go all the way because the gate is blocked. I told you there are 2 channels. This enzyme is fascinating. It's even more clever. It also regulates or communicates between these D channels through which the protons can go and the other channel somehow. electrostatics. I can give this away, don't have a slide on that. They somehow manifest in the lifetimes of hydrogen bonds between this asparagine and water molecules. Why are they important? So only if you have a hydrogen bond between the asparagine and the water molecule, so if that is long enough, that means the water molecule has long enough time to stay close to actually allow this passage. The lifetimes of hydrogen bonds depend, of course, on the protonation state in the D channel. So has the proton already arrived upstairs or not? But also on whether you have a protonated lysine in the other channel, the name-giving lysine. If you don't have, there's a very short lifetime for these important hydrogen bonds, or even if the proton still has to go through. So this is this situation, D1 meaning proton is still sitting here and not over there, and this lysine not having a proton. However, if channel 2, which is operative later, has a proton at the lysine, then we have a long enough lifetime for protons to make all the, well, sort of long enough lifetime for these hydrogen bonds, which should allow the arrangement of the hydrogen bond chain such that the proton transfer can occur later, as opposed to once the proton has arrived, we have a short lifetime. So the D channel feels, so to speak, that something is already going on in the K channel. Like, before the K channel does do its job, we have to be quick and 2 hours. Sort of. So the gating role of the asparagine is further controlled by the lysine and the communication between the 2 channels. And also K channel is something we looked at. I'm very quick with that. The K channel also auto-regulates in a bit more, not more difficult manner. What I'm showing here is snapshots of simulations of now different protonation versions in the K channel. Again, classical, proton put on a water molecule in the lower part, water molecule in the middle part, a water molecule, proton on this famous lysine, water molecule in the upper part. And you may or may not see on these snapshots that, again, the hydration increases with the proton moving up, number of water molecules. You may see it a bit better in these cloud pictures, upper row here. So when the proton is still in the lower part of the K channel, now the other channel, there is a sort of desert in the middle part, which also manifests in the hydrogen bond connections. So there are connections down here, but nothing allowing the proton to go to the upper part. We need hydrogen bonds for the proton transfer. If the proton moves up, there are more water molecules, so the channel becomes more and more wet. It's attracting waters with it. And now it's sort of not fully connected yet. If the proton is at the famous lysine, the channel is full of water and the connection is there. So hydrogen bonds from the lowest to the upper part, proton transfer is possible. And once the proton has arrived in the upper part, like here, hydration decreases again. You again have the desert. So proton can't move down again on no backlead. So the hydration increases with the proton moving where it should go. The hydrogen bond connectivity increases, everything fine, until the proton has arrived, and then it can't go back anymore. How does this happen? With closer inspection of having this full hydration, but the proton again put at different positions in the channel, and now having the lysine in 2 conformations. One is pointing down to the entrance of the channel, so it could grab a proton, and the other is pointing up, so it could donate a proton. So it's not the hydrogen bond, it will become the hydrogen bond connection, but it's the conformation of a long lysine going this or that way. The simulations are rather short, not to allow the lysine to do too much. Water molecules are more or less where they should be. These are the positions of the water molecules. These lines indicate different height levels in the channel by the position of important residues. And you see it's a bit more diverse here in the lower part where the water molecules are going up and down. And in the upper part, they stay clearly at the position they started with. More importantly, if you now compute the probability for proton to be in this water molecule or on that water molecule. That's been done by metropolis criterion. So in Monte Carlo, I think you've done that. Just comparing the energies, plus a boundary condition subject to, so allowing this exchange of proton being here or there, subject to there must be a hydrogen bond connection between the here and the there, because otherwise we won't have a proton transfer. We get the following out. Complicated. We see this is the position of the source water molecule, the protonated one, of the target one, with lysine pointing up, with lysine pointing down here. And the length of path is how many water molecules are in between. We see several things. We see that the paths are shorter once the lysine is pointing up. The length of the path is in this direction, so they are up to 6, whereas you have here even longer path. The positions where they go is mainly up once the lysine is pointing up, so remaining in this upper bit. Some are going down, of course, but whereas when the lysine is pointing down, you see long path in either direction, and you also see more scattered arrival zone of where the proton can go by the height of this. So everything is full of accepting water molecules, so to speak. This is how to interpret this picture. So diverse path lengths can go up, you can go down, as long as the lysine is pointing down. It can basically only go up and remain up when the lysine is pointing up. And this can be understood again by the hydrogen bond network and the directionality of which, this is a busy slide, I'm sorry for that, showing 3 examples. We have to read this now from the bottom to the top. So this is a situation where the lysine is pointing down. Proton is sitting, let's say, here. You have a connection and the arrows are pointing up. So hydrogen bond network is like, okay, proton has to move there. If the proton is a bit up in the channel, it's even more so. So like, okay, we also know that the hydration level hasn't changed in this simulation. It's the same number of water molecules, just for comparability. But the directionality of the hydrogen bond network is now even more to, we want. And with the proton sitting at the lysine, in the down conformation, now everything, well, it points down and up. You see arrows going down, you see arrows going up. So the proton could go either way. No regulation yet. Everything's connected, proton could go either way. However, if the lysine swings up now once it's protonated, and it does do that only with a decent probability in a protonated state, we have this situation here. Now, there is no connection between the upper part where the proton already is and the lower part in terms of hydrogen bonds. There's enough water molecules, but the directionality of the hydrogen bonds makes sure that the proton remains in the upper part and doesn't go back. Similar directionality we also see still when the proton would be lower in the upswung part. So even if the proton manages to come down, everything is pointing upwards again. So to summarize this busy slide, there's a weak connection between the upper and the lower part once the lysine has swung up, but that makes sure with the separation that there is no proton going back, no back leakage, and also no second proton coming once one proton is already where it should go, in the upper part of the channel. Summarizing the CCO part. So, taking together what I showed you today for cytopsia oxidase. In the detail, the gate opening of the asparagine is crucial, made us understand, so we could understand the proton transfer mechanism, we could understand how this is regulated because we found this asparagine gate to be important and to be regulated by the position of the proton being down or being up of the channel. So the protonation of this upper residue regulates whether the proton can still go through or not. We understood that in the other channel also the number of water molecule depends on the height of the proton, so whether it's already, but it's a bit more complicated because it also involves the conformation of this middle residue, the lysine pointing up or down. So that has a gating role here by its conformation. And in both cases, and there is communication within the channel, with a D and with a K channel, and between the two channels, and in both cases, the CCO channels regulate themselves, or the proton position makes sure that the second proton is not coming. Fascinating. Okay, I'm done almost. So I showed you very briefly something about pH-dependent photoexcitation. So we can calculate those things by combining classical MD and quantum mechanics and excited state calculations. I showed you very extensively something about DNA repair. We could understand how thymine DNA glycosylase recognizes, reacts, and not regulates. I showed you in the proton transfer example very briefly how with transition networks we could figure out the best pathway for the proton transport. I showed you communication, well, not pathways, but that there is something going on within the channels via hydrogen bond networks, something between the channels, and that there's a coupling of the reactivity, so the proton transport, water molecule numbers, and the conformational dynamics, which in the end gives the regulation or auto-regulation of cytochrome C oxidase. And I would like to finish with the most important slide. I wouldn't be here without those people shown on these 3 different pictures taken at different times from my group. I hope everybody who contributed to the stuff I showed today is on this. This is from Berlin. These are 2 pictures taken from Erlangen. We had collaborations in these projects with the people in New York, in Barcelona, in Berlin, of course. We used an awful lot of computational budget in Berlin, in the States, and nowadays NHR at FAU, that's Erlangen, the National Computer Center. We needed money. Almost all the money came, well, most of the money came from the German Research Foundation, some from the DAAD, some from a foundation in Berlin. So I'm very grateful to those people because they are the ones which made sure, or well, without them, you wouldn't have me here today. Neither featured or non-featured, not at all. And I'm grateful to you, of course, to invite me. And thanks again for the award and for your attention.
So now it's time for questions. Actually, I have one. In the DNA repair, you show a mechanism with 18 kcal and another one with 12.
Yes.
And I need you to explain me again this connection between them because I don't know if it's clear for me.
It was very fast. Yeah. Okay. So the upper round is the one mechanism for 18, the lower is the 12, the upper is the direct mechanism where the proton is sitting on this famous histidine, but it's not being transferred. The lower route is the proton is transferred to the base and then facilitating the dissociation, hence we have a lower barrier. The lower one is still the more probable one because it has a lower barrier. The lower one is the one which doesn't agree so nicely with the experimental barrier. So this is the, well, preaching bit here, word of caution. So do not compare just one number and be happy. There may be a pathway which doesn't agree so very well, but if you do the thinking, or if you are clever enough, you find an excuse, you can put it like this, and say, okay, what are we talking about? We're talking about probabilities throughout. So histidine has a certain probability of being protonated or not. That's encoded in the pKa. The lower the pKa, the lower the probability of being protonated. And well, with 7 point something, it's more than the probability of histidine in water. So the chance of histidine being protonated is already there, but it's not always protonated. And if you then, I didn't do the math here on the slide, and I don't remember the exact numbers. If you now calculate the conditional probability of going through the 12 or the 18 kcal pathway with a protonated histidine compared to the probability of having an unprotonated histidine and going through 30 kcal pathway, it boils down to roughly this 20 kcal actually. So it's a bit more involved.
Thank you. And also, you have already studied also these mechanism with the other system, or you have not?
Others were
Already.
faster, I would say. Yeah, we have, but well, maybe we still write a paper on that because we do not fully agree with the published mechanism. It's still dissociative, and according to another group, histidine is not important, which makes sense because there is not easy proton transfer to a cytosine, as I claimed on one earlier slide. Still, it is possible to different sites. So we're still not sure. Our numbers indicate that, again, proton transfer from histidine to another site on the cytosine base would help. But we're not finished with that, so I can't fully disagree with others. So the published mechanism is it's nice, it's fine. I'm not sure it has a full picture. So I don't want to belittle the work of others. So they did a great job. Okay, thank you. You're welcome. Please.
First, I want to say I really like the wide variety of topics you explained today.
Thank you.
I find it really, really interesting. And I want to ask you about how you decide which topics you want to study. Like, how will you reach the conclusion? Well, I think that's really interesting to study it by theory.
That's a very good question. I find so many things interesting and fascinating. It's more like, okay, what do I not work on? The DNA repair bit is something I really just discovered. So I did something on enzymes working on DNA before, and then I found the DNA repair interesting by chance. I found something in the literature. The proton transfer example is yet the opposite of this. So I was in Berlin in an initiative starting proton transfer, protonation dynamics. So that was brought to me, like, okay, we're doing experiments here, we're doing these kind of things. Would you like to contribute into calculations? And I was first hesitant to work on cytochrome C oxidase because it's such an awfully complex and difficult system. And it was a hate-love relation for a long time. I think by now it's more love than hate. There's still some, if you talk to people in the group of previous members, that, oh no, CCO, don't take me low. But so it's other side. The fluorescent protein was both. Initially, it started by me reading a paper on that. I said, oh, cool, I've done spectroscopy in my previous life, so to speak, and why not combine proteins with that? And then much later, very much later, and that's why we're still again working on that, I've been approached by a colleague who does experiment on that. Hey, you've published this paper on USFP, would you like to do something together? So, yeah, no recipe for life as a scientist here. If you can afford it, or if you get the money, do what you like.
Yeah.
Continue talking about the cytochrome C oxidase on the hydrogen path.
Mm-hmm.
You talked about the complex of choosing of the corrective variables to study the different movement. How did you bring it like this?
So we didn't. That was the trick. We didn't do a collective variable, one collective variable, but it's essentially it's a different variable for each of those mini connections here. So this is just schematic. So from one to the other, or maybe I'll show you the results. Yeah, because that's also going to, so say from here to this is already simplifying things. It's not a proton transfer so much, it's side chain rotation. From this to the next, it's now water moving. From this to the next, it's another water moving, and so forth. Because we couldn't say, okay, it's just pulling one. We could have pulled one charge, but I think we wouldn't got the right picture.
More questions?
Second one. I have two. So one regarding the top. In this proton transfer mechanism, so they're using full classical energy, right? So
this is QMM.
this is QMM, but the one
in the K channel, this was this was just classic.
Okay. So do you think maybe it's they are not relevant? But do you think that since you are investigating proton transfers or very light particles, handling effects are relevant?
Not at physiological temperature.
Okay.
We looked at that, not with a full quantum dynamics and everything, but we did a quick estimate with the WKB theory, so taking the barrier and then computing the tunneling probability. I don't know if you have. I mean, you know the tunnel effect, yeah? So you have the height and the shape of the barrier, which we do from our pathways, and you can estimate roughly the probability for the proton to tunnel, which would be relevant at very low temperatures, indeed, but not at 300K.
Okay.
It doesn't happen.
And maybe a question which is the answer would be interesting for our students, because they are having a lot of lectures in, I don't know, quantum dynamics, molecular dynamics, LVFT and everything. But we discussed yesterday that maybe in the future, the machine learning algorithms will be able to predict everything.
Yeah.
So why should we study all these computational approaches? So do you think it's worth to study?
It definitely is. First of all, it's fun. I don't neglect that. And the other is you understand much better. So you get insight. So of course, you can get insight also from a machine learned or predicted model. So if the model predicts you one protonation state is more probable than the other one, great. It's maybe the more tedious way, my opinion. So by having a physics-informed learning or a human learning, let's put, as opposed to the machine learning, by looking at all those things, you understand much better the physics behind the chemistry which is going on. Of course, since if you ask me my opinion, use as many tools as you can like. We're cheating here. We do QMM. We're not doing full QM of a protein. We use a rather cheap QM method just to be able to sample. We have a little bit of cheating, so to speak, or a bias by not testing every protonation possibility, but already have a certain bias. Although, and we use all sorts of tricks in the toolbox for enhanced sampling methods to make the calculation more quick. Why not generate the trajectories, let's say, by prediction, if you can have this with a pressing one button thing? Why not? And then make sure you still use your brains and, okay, what does it actually mean? What have I put into? What is the magic of the machine learning doing for me? Does it make sense? What has it been trained on also? It's the same as always questioning, so these force field methods, are they actually good enough? Is my 18 kcal barrier transition probably the one which is going on and not the rest? So my answer is no. So the physics tells me within my model, of course, the lower pathway is more probable. I could say maybe the whole QMM is wrong. We still don't know. We never know. But yeah, so just accept it. Well, take it as a tool, but don't be careful with this. Don't if they predict you. I mean, nowadays at least the predictions are made on training data, and if the training data doesn't contain, or is this too far from what you asked the thing to predict about, your prediction is not very good. A good model will tell you how bad it is. So something about the confidence or so.
More questions? Okay, so let's thank Petra for being here.
Thanks to that.
Okay, so thank you very much, Petra, for accepting again our invitation, for coming, and for the fantastic talk. It was amazing, all the things you can do with in classical molecular dynamics and also with QMM. So now we have a break, and we will have more time to discuss with Petra. Let me say again, Carmen, I will not forget that. Carmen, Sujel, thank you for the introduction, and thank you for the work to everyone. And so it was very nice to have you here, Petra.
A pleasure.
Thank you.
Thank you.